Let's switch gears here and start talking about the general categories of pathogens that we encounter and that are the cause of infectious disease. Let's talk about different types of pathogens and where those pathogens came from. First, we have three terms that describe the general type of a pathogen, and those terms are obligate, opportunistic, and accidental. An obligate pathogen is a pathogen that must cause disease. Its life processes are those that result in disease. Opportunistic pathogens are different. Opportunistic pathogens are organisms that can be transmitted without causing disease, but that under certain circumstances in the host organism, the obligate pathogen can take advantage of those circumstances and cause disease. And finally, we refer to some pathogens as being accidental pathogens. These are organisms that normally don't cause disease in the host. They only accidentally cause disease when they get into parts of the body where they don't normally reside. So obligate pathogens have to cause disease. In other words, they have to infect the host and grow and divide and damage tissue in order to survive and get transmitted on to new hosts. We described viruses as being obligate intracellular parasites. There are bacteria that are also obligate pathogens. The bacterium that causes tuberculosis is an obligate pathogen. The organism that causes syphilis, called Treponema pallidum, is an obligate pathogen. It has to infect the host organism and it will cause disease in order for the syphilis microbe to be transmitted from person to person to person. Opportunistic pathogens are different. They are taking advantage of a set of circumstances in the host where the host is not quite as able to fight off disease. A weakened immune system, for example, an altered microbiome, for example, a breach in the normal epithelial barriers. If you think about some concurrent diseases, skin conditions that might lead to excoriations and open areas, cut areas of the skin, burns on the skin. There are pathogens that take advantage of those kinds of breaches, those kinds of weaknesses in the host organism. And finally are the accidental pathogens. A good example of an accidental pathogen is one called Neisseria Meningitis. This is actually an organism that lives in the nose of a certain percentage of people, and it doesn't cause the host any problems at all when it's inside the nose. But there are times when the organism gets way to the back of the nose, and for one reason or another, the meninges are inflamed in the host, and that organism gets through the meningeal barrier and causes meningitis. So it's an accidental meningitis. It's not that the organism needs to invade the meninges in order to divide and grow and survive. It's just that it accidentally gets into the meningeal tissues, and once it's there, it grows and divides. Another good example of an accidental pathogen is one called Bacteroides Fragilis. This is an organism that normally lives in the colon and doesn't cause any disease. But it can accidentally lead to disease if it is released out of the colon. So during surgeries on the colon, or during trauma, if the colon is perforated, if Bacteroides fragilis gets out into the cavity of the abdomen, it can lead to peritonitis and significant sepsis. So it's interesting to ask the question about the source of all of these infectious pathogens that human beings and other organisms deal with. Where do pathogens come from, in other words? Now, there are times when pathogens arise from other organisms that are perfectly normal and even helpful, organisms of our microbiome, for example. So sometimes normal flora, what we call commensal organisms, can go through processes where they develop new virulence factors and become pathogenic. Obviously, mutations are behind the development of any new trait. But if the trait happens to be a new virulence factor, an organism that was once quite normal and even helpful to the host can suddenly become pathogenic. Now, these kind of events take time. They don't happen overnight. This kind of sort of evolutionary change takes time. And the only good news when a commensal organism becomes a pathogen is that it used to live in the human body. It's an organism that the human immune system has encountered before. Remember, we said part of the immune health of an individual comes from the fact that the immune system learns how to tolerate the organisms that belong in the body and not overreact to them. But part of that tolerance comes from the immune system recognizing which organisms are commensal. So there is some memory to a pathogen that arises from a commensal organism. The immune system knows that organism. It recognized it and it tolerated it when it was commensal. Other pathogens are said to be zoonotic in origin. In other words, they came from a different animal source. This is an instance where a pathogen originates in one particular animal species, goes through some mutation events, and develops the new trait of being able to invade and cause disease in a human. So any pathogen that we can trace back to a different animal source is said to be a zoonotic pathogen. Good examples of zoonotic pathogens are the virus that causes AIDS, the influenza virus that is known as the bird flu, that's H5N1, and of course our current pandemic coronavirus, SARS-CoV-2. All of the evidence that we have to date suggests that SARS-CoV-2 originated in a bat species. It underwent some mutation events and it developed a new trait of being able to not only infect humans, but to cause this terrible disease called COVID-19 in humans. Now, the final term that you'll hear in relation to sources of pathogens is this term novel. In order for a pathogen to be called novel, it has to be changed enough in its genetic material to be considered brand new. So new genes through mutation, giving it new traits, and giving it enough new traits so that there's no immune memory in the host organism. Now, the SARS-CoV-2 virus is also a novel pathogen. It arose from another coronavirus that lived inside of a particular bat species. It went through mutation events that changed it enough so that it could invade humans and develop disease in humans. But the reason we can call it novel comes from the fact that those mutations have changed that virus so much that it's essentially brand new. It is a version of the old virus that is so changed that it's essentially new, and human beings have no immune memory to it. So you could say that the SARS-CoV-2 virus is zoonotic and it's novel. Now, the other thing we need to think about when we talk about pathogens is where those pathogens reside and what other organisms might be involved in their transmission. Remember, while there are some pathogens that normally live inside humans, for example, and don't cause disease until a certain set of circumstances arises, I'm thinking about opportunistic and accidental pathogens. There are other pathogens that are coming into the human body from somewhere in the environment. We have to understand where the reservoir is, where the sort of the storage location is for a pathogen before they come into the human body and cause disease. So some pathogens, the reservoir is human beings. But for other pathogens, there are other animals where that pathogen normally resides. Sometimes pathogens live in the soil, or they live in water. They might reside on plants. Again, those places and those organisms would be considered the reservoir for that pathogen. And the reservoir is then a source of infection for any susceptible hosts. So reservoirs can be living or they can be non-living, like soil, water, etc. Now, when it comes to the reservoir themselves, living reservoirs don't get the disease that the pathogen is causing, or in some cases, they're experiencing a subclinical disease. They're experiencing something that is below what we can detect. Let's look at some examples of this. The reservoir for Lyme disease is actually pretty varied. It's several different small mammals, and it's several species of bird. So those animals, those small mammals and birds, are a place where the Lyme disease bacterium normally resides. But when ticks bite those animals, those small mammals, those birds, the tick can pick up the organism and then pass it to us when we get bitten by the tick. So the reservoir animals, they're not getting Lyme disease, but we are. We are the host, the susceptible host for that organism. There are some pathogens that have human beings as their reservoir. For example, polio and cholera are two terrible infectious diseases that have human beings as the reservoir. In other words, there's no other place in the environment, there's no other animal or plant or source of the pathogens that cause polio, the pathogen that causes cholera. There's no other place where they reside. They only reside in human beings. They cause disease in some human beings, but not in others. The reservoir for the pandemic virus SARS-CoV-2 is also humans. In other words, this virus in its current form can no longer survive and multiply and grow in bats. It can only do that in humans. So the reservoir is humans. And yes, there are humans that get disease from this virus, but there are other humans who don't, who just walk around carrying it, transmitting it, but not getting disease. So it's important to remember that this word reservoir is very different from the term carrier, because a carrier for a pathogen is an individual. A reservoir is an entire group, an entire species, or an entire genus, or an entire environmental location, like soil or water. Now, I mentioned ticks a minute ago, and ticks being involved in the transmission of the bacterium that causes Lyme disease. Any living thing that can transmit a pathogen from place to place is called a vector. So a vector is a living agent that carries a pathogen inside of them and transmits it to other susceptible host organisms. The vast majority of vectors are insects. Mosquitoes, fleas, ticks, mites, lice. If it's a little annoying insect species, it can also be a vector for infectious disease. There are other things that help spread pathogens that are not alive, though, and those are called fomites. So while vectors are living things that carry and transmit pathogens, fomites are inanimate objects that harbor and then transmit pathogens. So if you think, for instance, about countertops or workstations, surfaces that get touched, surfaces that come in contact with people all the time, with our hands and with our breath, and other secretions that come out of a human body. Things like syringes are fomites. Think about how a syringe is used. The needle is placed into one person's body, comes in contact with tissue and blood, and that syringe is now a fomite, because if the person it was injected into, if that person was carrying a pathogen, that pathogen is now on and in the needle, on that syringe, or in the syringe itself. That's a fomite now. Dental tools are fomites. Surgical instruments are fomites. Even everyday items like nail files and trimmers and brushes and combs can serve as fomites. Doorknobs, handrails, light switches, the keyboards on publicly available computers, exercise equipment at the gym, even things like bedding, blankets and sheets, hats, shoes, in some instances, even things like cardboard boxes can serve as fomites. So we have to know that fomites exist. We have to know how to disinfect fomites, and that will help in lessening the transmission of dangerous pathogens. Now, when it comes to fomites, it's also important for us to understand that different microbes can survive on these inanimate objects for a limited period of time. There was almost a degree of panic in March of this year when the pandemic first started to ramp up in our country and people began isolating themselves at home and quarantining at home. People were panicked about whether or not the objects they were touching were safe or were possibly a source of the coronavirus. People were concerned that the groceries they were buying might have viral particles on them, or that the boxes that were being delivered to their door might have viral particles on them. And the truth is that the SARS-CoV-2 virus, while it can survive on surfaces for a short period of time, it has not turned out that fomites are a big source of spread. We know now from the fact that so many people have had this virus and come down with COVID-19, we know that the greatest source of spread is respiratory droplets that are delivered from one person to another. You can pick up viral particles on surfaces, but for this particular virus, the spread through fomites is just not as important as the spread person to person. Now, what is it that allows a microbe to survive for a long time on a fomite versus another microbe not being able to survive? Well, oftentimes it depends upon the virulence factors that that particular microbe has, but it's also a factor of the things that those fomites get exposed to. So we know, for example, that desiccation or drying out is very detrimental to living cells. So bacterial pathogens are not going to be able to survive very long on a fomite that is very dry. On the other hand, fomites that are constantly damp, constantly wet, would be a good source for the survival of bacterial pathogens. The other condition, the other primary condition, is temperature. Any fomite that gets exposed to extremes of heat is probably not going to carry pathogens for very long. Now, there are pathogens, as I said, who have the right combination of virulence factors to allow them to survive on fomites for extended periods of time. And so those fomites and those pathogens can be, unfortunately, good sources for the transmission of infectious disease. The most common structure that can survive for the longest period of time on fomites are endospores. Remember, there are two genera of bacteria, of pathogenic bacteria, that can sporulate: Bacillus and Clostridium. And if those spores get onto fomites, they'll stay on those fomites essentially indefinitely until they gain re-entry into a host where it's warm and wet and full of lots of good nutrients, and the conditions will be right for that spore to revert back to the vegetative cellular stage and cause disease. I mentioned norovirus in a previous slide. It's been shown that norovirus can survive up to 4 weeks on certain surfaces. Remember, we said 14 viral particles are enough for the ID50. That's a scary virus, and you can understand why when norovirus breaks out in places like cruise ships, it's very easy for the virus to spread all the way through the population and to cause significant disease. Another scary example is the virus that causes hepatitis C. Drug users, particularly intravenous drug users, are plagued by hepatitis C. And experiments have shown that the hepatitis C virus can live in plain old drinking water for up to 7 days. So if you think about what an IV drug user does when they are mixing up drugs for injection, they often add water onto the dried drug in order to make a solution that they can inject. And unfortunately, a lot of IV drug users share, a bottle of water, and they're all drawing water out of it. And because the syringe and needle are fomites, the water becomes contaminated with hepatitis C. And that virus can survive in that water up to 7 days before it degrades. So again, some viruses can survive on fomites for long periods of time. So when it comes to the transmission of pathogens, the most successful pathogens, the ones that cause the worst kinds of infectious diseases, are the ones that can sporulate. Remember, sporulation produces that dormant form of a microbial pathogen that we call a spore or an endospore. The Bacillus species and the Clostridium species are capable of doing this, and they are thus capable of causing some very significant infectious disease. For what we understand today from all of the data and all of the experiments that have been conducted, bacterial endospores can survive indefinitely on fomites. They can survive in the absence of water. They can survive in the absence of nutrients, during extremes of temperature, during harsh conditions. They do not metabolize, and they're just inanimate forms of the pathogen. But once those spores get into a good environment, particularly a host organism, the conditions are ripe for them to return to the vegetative state, to become cellular once again, and to cause disease. Clostridium tetani causes the disease known as tetanus. Clostridium botulinum causes botulism. Bacillus anthracis causes anthrax. These are very bad pathogens, these sporulators. Very bad pathogens that cause very bad disease. The last source of pathogenic microbes that we'll talk about today are what we refer to as nosocomial. That word nosocomial just means hospital acquired. So there are some infections that get transmitted. There are some pathogens that get transmitted in the hospital setting. And it's become very problematic for hospitals to deal with these nosocomial pathogens and the infections they cause. Unfortunately, when people go into hospital for one disease or one problem, they can come out of the hospital with a brand new problem, which is an infectious disease. Currently, the data shows that there are between 2 and 4 million cases of nosocomial or hospital-acquired infections each year in our country. And those infections lead to somewhere between 20,000 and 40,000 unnecessary deaths. The typical pathogens that are nosocomial, that are causing nosocomial infections, are opportunistic pathogens and obligate pathogens. They get into the hospital, they're difficult to eradicate, and they infect these people who are vulnerable because they're in the hospital. They have some problem going on that has weakened their defenses. The other issue that has arisen in recent years is that a lot of these nosocomial bacteria are antibiotic resistant. And that's because, living in a hospital setting and interacting with each other, these cells interacting with each other, they are transmitting plasmids to one another horizontally. Remember, we said bacteria are capable of sharing plasmid DNA, and some of those plasmids carry the genes that make proteins that allow these bacteria to be resistant to our common antibiotics. On this slide, we can see a table that comes out of a textbook showing the body site distribution for nosocomial infections. On the left-hand side, you can see the different types of body sites, and on the right-hand side, you can see the percentage of all nosocomial infections that occur at each body site. You've got urinary tract through urinary catheterization, surgical sites, particularly sites of intestinal and joint replacement surgeries, the lower respiratory tract through the use of respirators and other breathing aids, and finally blood infection or bacteremia, and skin infection and other sites. On the right-hand side of the table, you can see that by far the urinary tract is the site of the most nosocomial infections that are encountered in hospitals. Approximately 50% of all nosocomial infections result from urinary catheterizations. When a patient comes into the hospital and for whatever reason requires the placement of a urinary catheter, that catheter is allowing certain pathogens to get into their bladder and into their body because it's open to the outside world. So, of course, urinary catheters have to be very carefully monitored and taken care of while they're used in patients in order to try to prevent nosocomial transmission. The last thing we're going to be talking about today is the transmission of pathogenic microbes. On this slide, you can see a comparison between what's known as direct transmission and what's known as indirect transmission. When a pathogen is transmitted from host organism to host organism, that is called direct transmission. When a particular pathogen is transmitted from a host organism to a vector or a fomite, and then to another host organism, that's referred to as indirect transmission. So, with regards to direct transmission, there are several ways that it occurs. First is just plain old physical contact, and that may include touch. It might include spreading saliva. It might include the transfer of blood or sexual fluids. Bites are a way for direct transmission to occur. That can be human to human bites or animal to human bites. One you may not think of right away is transplacental transmission. That's a form of direct transmission from mother to developing fetus through the placenta. And finally, there's aerosol. Now, I've got a star next to aerosol because this is a term that's received some attention lately. There are different words that are used to describe this kind of transmission, including things like airborne or respiratory droplet. There are slight differences between those terms and what they technically refer to, but the idea is the same. If a host organism is spreading material through their mouth, so they're coughing or sneezing or singing, and they're spreading respiratory type materials out their mouth and their nose, and they are then transmitting that respiratory material to another host, that's considered direct transmission. Now, some examples of indirect transmission. Some pathogens are transmitted through food and water. So the host that is infected with the pathogen contaminates the food, contaminates the water, and then that food or water is consumed by the next host organism who becomes infected. Fomites, of course, as I mentioned, are an important part of indirect transmission. Touching surfaces, doorknobs, keyboards, sharing utensils, eating utensils, drinking vessels, cups, and things like that, bedding, toys, even clothing. Those are the kinds of fomites that can spread an infectious pathogen from person to person through indirect transmission. And of course, vectors are responsible for the transmission of certain pathogens. So pathogens can be transmitted either directly from host to host or indirectly because of fomites and vectors. Regardless of the mode of transmission, though, pathogens all require the same thing. They need an opportunity to gain entry into the host. They have to have an opportunity to grow and multiply. And then they need an opportunity to get out of the host and go to the next host, essentially. So the other thing we need to consider when we talk about the transmission of pathogens is what's known as routes of entry and exit. Now, one of the most obvious routes in is injection. If you think about a needle, again, a fomite, a contaminated needle, if that needle is injected into the skin, then it is a route of entry into this new host organism. Scratches and bites and insect stings are also ways of injecting a pathogen into a host organism. So injection into the skin is one route of entry. Another obvious route is through the mouth. Now, the mouth can be a route into the gastrointestinal system. The mouth can also be one of the ways into the respiratory tract. When we talk about pathogens being transmitted from the mouth down into the gastrointestinal system, the term that we use for that is fecal oral transmission. If you think about a pathogen that affects the GI tract, those pathogens are being excreted into the fecal matter. And the way they are then transmitted to the next host is through the person who is infected not using the best hygiene after they use the bathroom. Fecal material, tiny traces of fecal material and the pathogen are then on their hands, and they then spread the pathogen, transmit the pathogen by touching things with their contaminated hands. So the next person who comes in contact with that contaminated hand or the surface that that hand has touched is going to pick up the pathogen on their hands and then introduce it to their own mouth just by touching their mouth. So the fecal oral type of transmission of pathogens is actually quite common, and that's because there are a lot of gastrointestinal pathogens. Now, we said that sometimes the mouth can serve as a route into the respiratory tract. The mouth, the nose, and the eyes, ocular, are entranceways to the respiratory tract. It became evident early on that the SARS-CoV-2 virus was gaining entry into the respiratory tract through the mouth, the nose, and the eyes. And some people that I know asked me, how can the eyes be involved with the respiratory tract? Well, if you remember from your anatomy, you have a lacrimal duct in the corner of your eye that transmits tears through the duct and out into the back of your pharynx, into the back of your throat. That's where tears drain ordinarily. And of course, the pharynx is essentially the entrance to the trachea, and from the trachea down into the respiratory tract. So yes, someone who is infected with certain respiratory pathogens, like the SARS-CoV-2 virus, can get viral particles in their on the surface of their eye, and those particles can then be washed through the tears down the lacrimal duct and into the back of the throat, and then be transmitted into the respiratory tract. It's also possible for the eyes to be a source of transmission for ocular pathogens. So, for example, if you think about pink eye is transmitted when the person who is infected and has the pathogen growing in their eye, on the surface of their eye, touches their eye, rubs their eye, and then passes the pathogen along to the next person who touches their eye and develops the infection. The gastrointestinal system can also be a source of infection for the urogenital system. Going back to bathroom hygiene, it's quite easy for pathogens that exit through the gastrointestinal tract, through the feces. It's easy for them to be moved manually to the openings of the urogenital tract while the person is using the bathroom and cleaning up after using the bathroom. And of course, there are some urogenital pathogens that are passed from one urogenital tract to another urogenital tract through sex acts, for example. And finally, the blood. The blood is certainly a source of transmission. We said that pathogens can be directly transmitted from a pregnant woman to her fetus through the placenta. And there are some pathogens, although not many, that can be transmitted from the vagina to the fetus through the cervix. That is fortunately an uncommon route of transmission. But during childbirth, as the fetus passes out through the vagina, if the vagina contains pathogenic microbes, it's possible for the fetus to pick up those microbes during the childbirth process. And there are some pathogens that we monitor for around the time of childbirth. So it's important to remember that pathogens have specific routes of entry. Sometimes there's a singular route, sometimes there are multiple routes. Pathogens are restricted, essentially, by what disease they can cause based on what tissue they're able to infect. So pathogens cannot cause disease outside of those tissues, outside of that route of entry, in other words. For example, a respiratory pathogen cannot cause intestinal disease. Now, yes, there are some pathogens that are capable of causing both respiratory disease and intestinal disease. But if it's a purely respiratory pathogen, like, for example, influenza, it's not capable of causing disease in another body system. In other words, there is no such thing as the stomach flu or the intestinal flu. Influenza is a respiratory pathogen. It can't cause disease in the intestinal tract. Just like intestinal pathogens can't cause skin infections, for example. It just doesn't happen. Pathogens are adapted to a particular type of tissue, and they're capable of causing disease in those tissues if they can gain entry to those tissues. It's important to remember, though, having said all that sometimes routes of entry can be connected in very mysterious ways. For example, imagine a respiratory pathogen. A person who's infected with this respiratory pathogen coughs into their hands, and their hands become covered in that pathogen. That person then touches a doorknob and deposits that pathogen onto the surface of the doorknob. Now another person comes along and touches the doorknob and picks the pathogen up on their hand. And then that person quite innocently rubs their eyes because they're tired. And they introduce that pathogen onto the conjunctival tissue in the eye. Tears from the eye wash the pathogen off of the conjunctival tissue, down into the lacrimal duct, back into the pharynx, and then from the pharynx down into the respiratory system, And now the pathogen is where it's capable of causing disease in the respiratory tract. I want to make just a couple of final points about the transmission of pathogens and the transmission of infectious disease. The transmission of a pathogen depends upon the biology of the pathogen. All the things that we've talked about in this lecture are really important biological factors for the transmission and occurrence of infectious disease in populations. But it's important that we remember that transmissibility of a pathogen is also impacted by things that have nothing to do with biology. I'm talking about cultural factors and socioeconomic factors. And we don't talk about these things in a course like this, but these are things that have to be considered by the people who deal with public health issues. Cultural factors are an important thing to consider. For example, in certain cultures, it's not normal to use silverware to eat. It's culturally appropriate to use your hands while you eat. And so you can imagine that a group of people who are sitting around a common source of food and who are using their hands to consume that food are more vulnerable to the spread of pathogens that are transmitted through food. There are certain cultures who practice bathing very frequently. There are others who practice it much less frequently. Different cultures have different hygiene practices, different sexual practices, and those practices can impact how likely or how unlikely it is that certain pathogens will be transmitted. The other factors that are important to consider are socioeconomic factors. And these factors have really come into play in the current pandemic. If you think about, for example, things like how many individuals live in the home and how big the home is. If there's a single person living in a 2-bedroom apartment in a city, if that person quarantines themselves in that apartment, that's a great way to reduce the transmission of disease to that person. But imagine if you have a family of 10 living in that 2-bedroom apartment. Now, simply quarantining those people into that apartment, first of all, becomes a lot more difficult. And second of all, does not keep those people far enough away from each other to prevent transmission of a pathogen. Things like whether or not people have access, easy access, ready access to clean drinking water matters. Whether or not people have the means to go out and buy disinfectants and cleaning products, the quality of the food they're eating, whether or not they're able to get regular medical care. All of these things impact how hard or how easy it is for a pathogen that causes an infectious disease to spread throughout a community. Both biology and these other factors have to be addressed when we put together public health programs in order to stop the transmission of these dangerous pathogens.